Hello, how are you all doing? In today's class, we will study sickle cell anemia. Sickle cell anemia was also taught in chapter number 5, Principles of Inheritance and Variation. This is actually the topic of chapter number 5, but at that time you were not knowing what are coding and template strands, so I am going to explain it here in detail. Sickle cell anemia is an autosomal recessive disorder. Okay, this is important to note that it is an example of autosomal recessive disorder. Okay, now when I say autosomal, means in the pedigree of this disease or ailment, almost equal numbers of males and females, they will be affected. When I am saying recessive, means it will skip the generations. Affected parents may have unaffected children and unaffected parents may have affected children. So this is the trick to solve the pedigree. For more details, you can check out the previous videos or I'll share in my I button. Okay. So sickle cell anemia is a disorder which happens on chromosome number 11. That means it is a disorder because of the autosomes, because chromosome number 11 comes in the category of autosomes. In sickle cell anemia, what actually happens is RBCs, they are normally biconcave in shape, right? And they have hemoglobin pigment inside them which carries oxygen. In sickle cell anemia, the RBCs, they become sickle shape. A person could be homozygous for this trait or it could be heterozygous. Homozygous individuals, I mean the people who carry two recessive alleles, because for this disorder to happen, a person should carry two recessive alleles. And such type of disorder will come when two heterozygous parents, they will marry each other. The probability of the sickle cell anemia in the offspring, it will increase. That's what we read in chapter number 5. So the people who suffer from sickle cell anemia, they have two recessive alleles. The carrier individuals, they will carry only one recessive allele. Okay, and the homozygous individuals which carry two recessive alleles, they die at a very young age because their oxygen carrying capacity is very less. So they cannot cope up with all such and they die. Whereas the carrier individuals, they live their normal life, but once they go to the high altitude, it creates problem in them. Now we will see what actually happens and what actually distorts or changes the shape of the RBC. Now you know that DNA is double helical. One strand is known as the coding strand and the another is known as the template strand. We have already done with point mutation, so sickle cell anemia comes under the point mutation. There is a sequence on the DNA which is GAG. Remember, hemoglobin is a 4 polypeptide containing structure. Means it exhibits quaternary structure because it is made up of more than one polypeptide. So two chains are similar to each other which are known as HBA and two chains are similar to each other which are known as HBB. So this I am talking about HBA chain, and there is a sequence which is known as GAG, and in the normal person, GAG will code for CTC in the template strand, and if we are going to make the messenger RNA, we know that the coding strand and the messenger RNA, they have the same sequence. And if I talk about the protein which is formed or the amino acid which is being synthesized by GAG, it is glutamic acid. So this is the 6th amino acid of the HBA chain. Now what actually happens in the people who suffer from sickle cell anemia? This A is being replaced by T. Okay, rest sequences, they will remain as such. right? So this sequence here in between, it has changed to T. Remember here A is an example of purine and T is an example of pyrimidine. So what substitution has been happened? Yes, you are right. This is transversion. This we have discussed in the point mutations, that whenever purine is replaced by purine or pyrimidine is replaced by pyrimidine, it is transition. But here what are we noticing? Yes, we are noticing transversion because purine has been replaced by pyrimidine. Now, as the template strand, in this case, it will have C, A, and C again. If you will talk about its messenger RNA, then its messenger RNA will have G. There is no T in messenger RNA. We will write U and GUG. So you can see here the codon, earlier codon was GAG and now the codon is GUG. So the codon has changed. Let's see what amino acid is being coded by this codon. So it is valine. Okay. Now earlier it was glutamic acid which was coded by GAG. Now it is valine. So amino acid has been changed. And this can also come under missense mutation, because in missense mutation the amino acid is changed. Okay, this is a point mutation. Why? Because there is a change in only one nucleotide. Why there is missense mutation? Because amino acid from then and now has been changed. Now this newly formed valine, it will disturb the forces acting in the hemoglobin molecule, and this is responsible for making the RBCs sickle shaped. So once the question on sickle cell anemia comes, you must know the genetics as it was told in chapter number 5, its pedigree. Problems on pedigree is also important, and the question may come specifically on the molecular biology, like explain the molecular biology of sickle cell anemia. So you need to say that sickle cell anemia is a genetic disorder in which problem arises in chromosome number 11. Chromosome number 11 comes in the category of autosomes. That is why it is known as autosomal disorder. It is recessive disorder too because it skips the generation. Then you can explain the way I have made this illustration for you. Similarly, you can make illustration and you'll be getting full marks. Remember, if the time is less in the exam and you are facing problem in completing whole questions, it is always better to draw the illustrations, because for illustrations also the examiners, they give the marks. But don't skip the question, because skipping a question will give you a straightforward 0. So it's always better to get few marks than not full. So draw the illustrations, write 2-3 sentences. There is a probability that you will get in good marks. I hope it was helping to you. If you haven't subscribed to my channel, please do so. Stay tuned for the next video. Happy learning.